Hello, my name is Mendoza, and this is my presentation for Exam 3, Part 3. So, in order to really answer the questions, we need to basically go through a little bit of an explanation of what DNA fragmentation is, apoptosis, and the DNA structure. So, one of the ways that DNA fragmentation can happen is whenever a cell goes through apoptosis and dies. DNA fragmentation is the separation or breaking of DNA strands into pieces. Apoptosis is a type of programmed cell death. Whenever the cell recognizes that has a mutated strand of DNA, that affects the cell negatively, it generally undergoes through apoptosis and sacrifices itself in order to not replicate the mutated DNA. Histones are some basic proteins that are found in eukaryotic chromosomes, and they include proteins such as H1, H2A, H2B, H3, and H4. Nucleosomes are structural units of DNA in which the histone proteins form. Proteins such as the H2A, H2B, H3, and H4 basically get together and form an octamer, which is basically two proteins of the H2, H2B, H3, and H4. And it's basically, they form the nucleosome core, which DNA wraps around it. Linker DNA wraps around each of these nucleosomes that stretch about 50 base pairs in length. The H1 protein binds to the linker DNA in order to facilitate the packaging of nucleosomes into fibers of 30 nanometers each, an average of 146 base pairs to 200 base pairs of DNA wraps around a single nucleosome. In the picture right here, we can see that the structure of the nucleosomes, which is basically two proteins of each of the H2, each of these individual proteins wrapped around the DNA linker, and right next to it, behind the actual structure, the H1 protein helps it stay together and stay linked. So why this pattern is created upon DNA fragmentation? So when DNA when DNA from a cell that went through apoptosis is run on an agarose gel, the fragmented DNA differs in size, but it basically creates a ladder-like appearance on the gel. It stains the gel in a way that it forms kind of like a ladder. For this to happen, first the ligands on the membrane bind to death receptors that trigger an initiator caspase. Then this caspase triggers the executioner caspase 3, and the activation of this caspase basically promotes the death of proteins that would counterbalance anti-apoptotic proteins such as BCL2, which stop the cell from going through apoptosis. Cytochrome C is then released from the mitochondria that then initiates caspase 9 and by binding to an apoptotic protease. This caspase then cleaves and forms the executioner caspase 3 that triggers apoptosis. During apoptosis, the nucleus fragmentation occurs. This happens when endonuclease enzyme which is enzymes that cleave DNA, cleaves at the end of linker DNA the linker DNA region between two of the nucleosomes. This produces the DNA fragments that are just about 200 base pairs. This is basically the cause of the ladder-like patterns on the agarose gel. As we can see from the figure, they're basically like curved segments that form like a ladder. So yeah, and the nuclease basically cuts DNA and makes smaller fragments that are basically laid in that ladder-like pattern. So one of the other biochemical hallmarks of apoptosis happens as blevings. So blebs are bulbs of plasma membrane cells that are going through apoptosis. During apoptosis, the cytoskeleton breaks up and bulbs bulge outwards. These bulbs separate and contain some cytoplasm and proteins. At first, the initial blebs are small and only contain like cytoplasm and proteins. Later, through apoptosis, the blebs are larger and contain larger organelles such as the nucleus. I found an article where they did an experiment that proved that apoptosis depended on proteins such as rock proteins, which basically rock proteins are Rho-associated protein kinase. Proteins such as rock are important proteins that allow the cell to go through apoptosis and create blebs. This proved that this was proved by scientists by creating an experiment where they inhibited cells going through apoptosis. This is where they got this figure from. They looked at a cell where they did not change anything and use that as a control group, and the other group of cells were transfected with siRNA to block the function of ROC1 proteins. The figure shows the two groups and their effects. The cell that was not interrupted was able to show blebbings all around the outside of the cell. The cell that was transfected, though, showed no sign of blebbing. This demonstrates that in order for the cell to eject and form blebbings, proteins like ROC1 proteins are essential for blebbing formation. Another hallmark of apoptosis would be the phagocytosis. So phagocytosis is a type of endocytosis, and where matter or other cells are taken up from the environment and they get digested by neighboring macrophages. These macrophages help remove pathogens and cell debris such as apoptotic cells. Macrophages that take up dying cells through phagocytosis this process is called efferocytosis. So during the experiment on the article that I found, they basically determined that the cleaning of apoptotic cells by phagocytic cells is important for protecting tissue from inflammatory and immunogenic contents of dying cells. This process basically releases anti-inflammatory mediators such as TGF-B1 and suppresses pro-inflammatory cytokines such as TNF-alpha. The clearance of apoptotic cells reduces the chance for tissue inflammation in the body. They looked at the way that phagocytes would recognize the apoptotic cells. They basically noticed that different receptors would interact with their ligands on the apoptotic cells, as you can see from the figure. They basically realized that some of the examples of these receptors are things like B2 GPI receptor, the MER, and PSR. After the absorption of the apoptotic cells by phagocytosis, they basically noticed that there was an increase in production of mediators such as the TGF-B1 and the PGE2, and the increase of these two mediators were associated with a decrease in production of the pro-inflammatory mediators such as TNF-alpha and IL-8. These are my references, and thank you.